Forensic biology is the application of biology to associate a person, whether suspect or victim, to a location, an item, another person. It can be utilized to further investigations for both criminal and civil cases. Two of the most important factors to be constantly considered throughout the collection, processing, and analysis of evidence are the maintenance of chain of custody as well as contamination prevention, especially considering the nature of the majority of biological evidence. Forensic biology is incorporated into and is a significant aspect of numerous forensic disciplines, some of which include forensic anthropology, forensic entomology, forensic odontology, forensic pathology, forensic toxicology. When the phrase forensic biology is utilized, it is often regarded as synonymous with analysis of biological evidence. Disciplines. Brief history of forensic science. The first known briefings of forensic procedures we still use today are recorded to be used as far back as the 7th century. The concept of utilizing fingerprints as a means of identification was first established. By the 17th century, forensic procedures were used to account criminals of guilt charges, among other things. Nowadays, autopsies and forensics have skyrocketed not only in popularity, but also in technological advances. One of the first men to use such techniques to what we know now as forensics was a man by the name of Alphonse Bertillon, who in 1879 became the first to evolve a scientific system of personal identification. Through the development of the science of anthropometry, which involves the series of measurements of the body to help in distinguishing one human individual from another. Another man who not too long after made significant discoveries in the field of forensics was a man named Karl Landsteiner. Karl Landsteiner in the year 1901 discovered that the blood could be grouped into different categories, A, B, AB, and O, which of course led to more studies and eventually a whole new spectrum in the field of not only medicine, but also criminology. Dr. Leon Lattice himself is also a very well-known man, who made significant additions into the field of forensics. In 1915, he discovered a simple procedure to determine the blood group of the dried blood stains. This technique was adopted for criminal investigation. Another man was H.O. Albrecht. H.O. in the following years, 1928, to be exact, was a German chemist who developed a chemical solution, that is luminol. The luminol makes the blood glow. It is useful in detecting the blood stains at the scene of a crime. Other more important and significant men, like Sir Alec Jeffreys, are the ones to thank, since they are the ones who have shaped forensics into what we know now. In 1984, he developed the DNA fingerprinting technique to examine the variations in the genetic code, which can be used to distinguish one individual from another. Carey B. Mullis was also another man who the world of medicine and criminology should not thank enough. Carey in 1993, he developed the PCR technique that was used to amplify the samples of DNA fragments in vitro. This technique was useful for amplifying the DNA sample obtained from the scene of a crime in an extremely minute amount, degraded state, and a mixture of body fluids of two or more people. This technique has also gone on to not only solve crimes, but also other things, like detecting diseases, viruses, and many other DNA-related subjects. DNA analysis. DNA, or deoxyribonucleic acid, is one of the most popular pieces of evidence to recover at a crime scene. More often than not, evidence containing DNA is regarded to as biological evidence. With all of the substantial advances that have been made regarding DNA, biological evidence is recognized to be the golden standard in forensic science. At the scene, biological evidence must be initially visibly recognized. Sometimes this is not always possible, and the aid of an alternative light source, or OLS, is required. Once identified as a potential source, presumptive tests are conducted to establish the possibility of the specified biological presence. If positive, samples are collected and submitted for analysis in the laboratory, where confirmatory tests and further tests are performed. DNA analysis has numerous applications such as paternity testing, identifications of unknown human remains, cold case breakthroughs, as well as connecting suspects and/or victims to a piece of evidence, a scene, or to another person. Nuclear DNA evidence can be recovered from blood, semen, saliva, epithelial cells and hair. Furthermore, mitochondrial DNA can be recovered from the shaft of hair, bone, and the roots of teeth. For most forensic DNA samples, STR analysis of autosomal short tandem repeats is performed in an attempt to individualize the sample to one person with a high degree of statistical confidence. STR electrophoregram of a 3-person mixture laboratory analysis of DNA evidence involves the sample DNA being extracted, quantitated, amplified, and visualized. There are several methods of DNA extraction possible, including organic extraction, Chelix extraction, and differential extraction. Quantitation is commonly conducted using a form of the polymerase chain reaction, known as real-time PCR, quantitative PCR, or qPCR. qPCR is the preferred method of DNA quantitation for forensic cases, because it is very precise, human-specific. Qualitative and quantitative. This technique analyzes changes in fluorescent signals of amplified DNA fragments between each PCR cycle, without needing to pause the reaction, or open the temperature-sensitive PCR tubes. In addition to the components necessary for standard PCR reaction, i.e. template DNA, carefully designed forward and reverse primers, DNA polymerase, DNT, PS, and a buffer solution containing Mg2+, qPCR reactions involve fluorescent dye-labeled probes that complement and anneal to the DNA sequence of interest that lies between the two primers. A reporter dye is attached at the 5 end of the fluorescent probe, while a quencher dye is attached at the 3 end. Before the DNA strands are extended by the polymerase, the reporter and quencher are close enough in space that no fluorescence is detected by the instrument. As the polymerase begins to extend the strand, the 5 end of the probe is degraded by the polymerase due to its exonuclease activity. The reporter dye is released from the 5 end and is no longer quenched, thus enabling detection of fluorescence. A graph is constructed for the sample DNA comparing the presence of fluorescence to a cycle number of the qPCR process. This is then compared to a standard curve of the cycle fluorescence threshold versus the log of known DNA concentrations. By comparing the sample data to the standard curve, one may extrapolate the DNA concentration in the sample, which is essential to move forward with PCR amplification and capillary electrophoresis to obtain a DNA profile. DNA profiles are produced in the form of an electrophoregram. The obtained profile can be compared to known samples such as those in CODIS in order to identify a possible suspect. Based on known frequencies of the genotype found in the DNA profile, the DNA analyst may place a statistical measure of confidence on DNA match. Mitochondrial DNA analysis. Mitochondrial DNA is used instead of nuclear DNA when forensic samples have been degraded, are damaged, or are in very small quantities. In many cases there may be human remains that are older, sometimes ancient, and the only options for DNA collection are the bone, teeth, or hair of the body. DNA is able to be extracted from such degraded samples because its presence in cells is much higher than nuclear DNA. There can be more than 1,000 copies of DNA in a cell, while there are only 2 copies of nuclear DNA. Nuclear DNA is inherited from both the mother and the father, but DNA is passed down from only the mother to all of her offspring. Due to this type of inheritance, DNA is useful for identification purposes and forensic work, but can also be used for mass disasters, missing persons cases, complex kinship, and genetic genealogy. As mentioned, the main advantage of using DNA is its high copy number. However, there are a few disadvantages of using DNA as opposed to nuclear DNA. Since DNA is inherited maternally and passed to each offspring, all members of the maternal familial line will share a haplotype. A haplotype is a group of alleles in an organism that are inherited together from a single parent. The sharing of this haplotype among family members can cause an issue in forensic samples, because these samples are often mixtures that contain more than one DNA contributor. Deconvolution and interpretation of DNA mixtures is more difficult than that of nuclear DNA, and some laboratories choose not to attempt the process since DNA does not recombine. The genetic markers are not as diverse as autosomal STRs are in the case of nuclear DNA. Another issue is that of heteroplasmy. Heteroplasmy is when an individual has more than one type of DNA in their cells. This can cause an issue in the interpretation of data from questioned forensic samples and known samples that contain DNA. Having adequate knowledge and understanding of heteroplasmy can help ensure successful interpretation. There are some ways to improve success of DNA analysis. Preventing contamination at all testing stages, and using positive and negative controls is a priority. In addition, the use of many amplicons can be beneficial. When a sample of DNA is severely degraded or has been obtained from an ancient source, the use of small amplicons can be used to improve the success of amplification during PCR. In these cases, primers amplifying smaller regions of HV1 and HV2 in the control region of DNA are used. This process has been referred to as the ancient DNA approach. The first use of DNA as evidence in court was in 1996, in state of Tennessee v. Paul, where there was only circumstantial evidence against Ware, so the admittance of DNA from hairs found in the victim's throat and at the scene were key to the case. In 2004, with the help of the National Center for Missing and Exploited Children in Choice Point, DNA was used to solve a 22-year-old cold case where the nuclear DNA evidence was not originally strong enough. After DNA analysis, R.B. Dean Williams was convicted of the murder of 15-year-old Linda Strait, which had occurred in 1982. In 2012, DNA evidence allowed investigators to establish a link in a 36-year-old investigation into the murders of 4 Michigan children. Hair fibers found on the bodies of 2 of the children were tested, and the DNA found to be the same for each sample. For the investigators, this was a big break, because it meant that the murders were likely connected. Forensic anthropology. Anthropology is applied to forensics most regularly through the collection and analysis of human skeletal remains. The primary goals of anthropological involvement include identification and aiding in scene reconstruction by determining details regarding the circumstances of the victim's death. In cases where conventional techniques are unable to determine the identity of the remains due to the lack of soft tissue, anthropologists are required to deduce certain characteristics based on the skeletal remains. Race, sex, age and possible ailments can often be determined through bone measurements and looking for clues throughout the skeletal structure. Forensic botany. Within forensic biology, we have the category of forensic botany. Forensic botany deals with the study of plants, leaves, seeds, and pollen or plant properties that can lead to traces of evidence in crime scenes that would be considered physical evidence. Before a plant can aid the investigations, the plant must be identified first to see if it is from the location that the plant was found. Forensic botany can help investigators by providing a link between an individual and the crime scene by pinpointing the geographical location of missing bodies or estimate where the burial took place. Another way that is used is through the interrogation process, where investigators compare the traces that the victim are found and compare to the statements of the suspect. Forensic botany can also reveal whether a death is due to suicide, accident, or homicide. Another example of forensic botany aiding investigators is by moss. Moss can establish the MI of a human skeletal through its growth rate. Example: an elderly man fell a steep hillside and was found dead. They concluded that the man did not die because of the fall, but from hypertensive heart disease. Forensic botany concluded that fall was approximately 10 m, and there was a lane that joined the hill path, around 9 m perpendicular from the body. It showed he had leaves on his left hand and sweater. There were also broken bushes in the scene. Forensic scientists who specialize in forensic botany had grabbed 5 soil samples and some plant samples from the scene. The plant samples were analyzed through optical microscopy, and they also examined the victim's clothing since it did carry some plant elements present. In this, they did a macroscopic and microscopic analysis which were compared to the collected samples on the scene. It's concluded that there was no evidence of struggling caused by another person. Instead, the elderly man had lost his balance. Subspecialties in forensic botany. There are subdisciplines within forensic botany. Examples include forensic palynology, dendrochronology, lichenology, mycology, and bryology. Palynology, for example, can produce evidence of decomposition time, location of death, or the time of year. Bryology is the easiest to find evidence since bryophyte attaches to shoes and clothes easily. Bryophyte useful since even if it is ripped apart or broken down, DNA can still be analyzed. Ways in DNA can be analyzed is through the microscope or other sophisticated DNA testing. Forensic ornithology. Bird remains can be identified first and foremost from feathers. Forensic odontology. Dentistry has helped law enforcement in criminal and civil proceedings by detecting and solving cases. Odontology did not get popular until the 1960s when an interest was brought up by the first instructional program in the United States at the Armed Forces Institute of Pathology. Because of this program, the term forensic odontology is now familiar in the dental profession and law enforcement agencies. Odontologists or dentists can use dental science to know a person's identity. Teeth can be used as weapons and therefore leave information about a person. This information is key in recognizing abuse among people of any age. Forensic pathology. A forensic pathologist is a medical doctor who is an expert in both trauma and disease, and is responsible for performing autopsies. He/she applies their extensive knowledge of the human body and possible internal and external inflictions as he/she performs an autopsy to hopefully ascertain the manner and cause of death. Information derived from the autopsy often greatly assists investigative efforts as well as scene reconstruction. Forensic toxicology. Forensic toxicology is the use of toxicology and other disciplines such as analytical chemistry, pharmacology, and clinical chemistry to aid medical or legal investigation of death, poisoning, and drug use. The primary concern for forensic toxicology is not the legal outcome of the toxicological investigation or the technology utilized, but rather the obtainment and interpretation of results. Forensic microbiology. With recent advances in massive parallel sequencing or next-generation sequencing, forensic microbiology has become an increasingly promising area of research. Initial applications in circumstances of biocrime, bioterrorism, and epidemiology are now accompanied by the prospect of using microorganisms as ancillary evidence in criminal cases to clarify causes of death, to assist human identification, for geolocation, and to estimate post-mortem interval. Bioterrorism and epidemiology. It is important to remember that biological agents that can be used as weapons are often found in the environment. For this reason, it is always difficult to determine whether infections associated with these bioagents are accidental or purposely started. While not the first or only incidence of bioterrorism, perhaps the most notable case in recent memory involved the sending of at least 4 anthrax-containing envelopes in the United States in September and October 2001. At least 22 victims contracted anthrax. As a result of the mailings, 11 individuals contracted inhalation anthrax, with 5 of these infections resulting in fatalities. Another 11 individuals suffered cutaneous anthrax. In addition, 31 persons tested positive for exposure to B. anthracis spores. However, thanks to advancements in PCR and whole genome sequencing, scientists were able to collaborate with the FBI and were able to identify the source of the letter spores. Post-mortem analysis. Post-mortem microbiology aims to detect unexpected infections causing sudden deaths. He/she applies their extensive knowledge of the human body and possible internal and external inflictions as he/she performs an autopsy to hopefully ascertain the manner and cause of death. Information derived from the autopsy often greatly assists investigative efforts as well as scene reconstruction. Forensic toxicology. Forensic toxicology is the use of toxicology and other disciplines such as analytical chemistry, pharmacology, and clinical chemistry to aid medical or legal investigation of death, poisoning, and drug use. The primary concern for forensic toxicology is not the legal outcome of the toxicological investigation or the technology utilized, but rather the obtainment and interpretation of results. Forensic microbiology. With recent advances in massive parallel sequencing or next-generation sequencing, forensic microbiology has become an increasingly promising area of research. Initial applications in circumstances of biocrime, bioterrorism, and epidemiology are now accompanied by the prospect of using microorganisms as ancillary evidence in criminal cases, to clarify causes of death, to assist human identification, for geolocation, and to estimate post-mortem interval. Bioterrorism and epidemiology. It is important to remember that biological agents that can be used as weapons are often found in the environment. For this reason, it is always difficult to determine whether infections associated with these bioagents are accidental or purposely started. While not the first or only incidence of bioterrorism, perhaps the most notable case in recent memory involved the sending of at least 4 anthrax-containing envelopes in the United States in September and October 2001. At least 22 victims contracted anthrax. As a result of the mailings, 11 individuals contracted inhalation anthrax, with 5 of these infections resulting in fatalities. Another 11 individuals suffered cutaneous anthrax. In addition, 31 persons tested positive for exposure to B. anthracis spores. However, thanks to advancements in PCR and whole genome sequencing, scientists were able to collaborate with the FBI and were able to identify the source of the letter spores. Post-mortem analysis. Post-mortem microbiology aims to detect unexpected infections causing sudden deaths, confirm clinically suspected but unproven infection, evaluate the efficacy of antimicrobial therapy, identify emergent pathogens, and recognize medical errors. Additionally, the analysis of the thanatomic may help to estimate the post-mortem interval. There is currently an extensive amount of research being performed, most notably using the famous body farms throughout the United States, to determine if there is a consistent microbial decomposition clock that could be used by itself, or in conjunction with other techniques to help estimate post-mortem intervals. One such group has made extensive headway into describing such a microbial clock, and believes she's within 2 to 5 years of testing her clock in a real crime scene scenario. However, if a reliable and consistent microbial clock is determined to exist, it's too soon to know whether the microbial clock will pass scientific and legal muster. And a judge would also have to determine that the microbial clock meets the standard for admission of expert testimony. Water sample analysis. In cases involving a body of water at or near the scene of a crime, a sample of the water can be extracted and analyzed under a light microscope for microorganisms. One such microorganism that are analyzed within samples of fresh water are diatoms, microscopic algae of varying shapes. Different bodies of water have been found to contain unique sets of diatoms, and therefore, a piece of evidence found in a specific body of water will contain unique diatoms on it found only in that specific body of water.